Right. So then see the muscle contraction. This chapter is also very much important. Hope you guys are knowing what is heavy chain, which is heavy chain, and which are the light chain. Okay. You know myosin, actin. Have you heard of these things? Have you heard of these terms? Yes. Hopefully you have heard of this. Okay. So see, these are the myosin. These are what? Myosin. This is myosin. And these are what? These are actin. Okay. So again over here, same way in this image, hope you can see clearly. The one in the middle, that is the myosin, and one in the over here, it is the actin. Okay. So first of all, in the past, they have asked to do the nomenclature. Okay. Tell me the line over here. So this is the Z line. Okay. See, each and every class we are going to study like this only. If a topic is something to be explained, I will make you teach like this. If it is like basically theory, we will study it from a particular literature. That will be directly through the literature. And someone is asking me that, sir, will we get the notes? Okay, if you want, yes, I can share you this. My OneDrive is there, OneNote is there, you can share it. But better than that is what you guys, all of you guys have that QRS. Yes, week revision, this thing. QRS is like volume 4. QRS volume 4 is available with all of you guys except 2-3 students. I have been explaining like that. So all of these students, you must keep your QRS with you while I'm teaching. You should not keep on writing. You should not keep on making the notes while you're studying, because this is rapid revision. If it is a general class, definitely I would have said you that make notes, make notes, make notes. At this particular given point of time, when exam is just 25 days away, you cannot just keep on writing. Now you have to revise. So keep your QRS ready and keep it beside you. Okay, some points we will be directly studying from QRS. Okay, I will be directly showing some pictures from QRS, because it has been tremendously designed and excellently made, and it has got clear strike rate of at least more than 70 to 80%. Okay, so that is high. That is how high these stakes are. Okay, all right. So let me show you. See, the basic unit of muscle contraction is called as sarcomere. So please remember sarcomere. Let me draw sarcomere starting from here to here. This part is called as one sarcomere. There is one more sarcomere here. This is the second sarcomere. So see, this is called as sarcomere. So it is the basic unit of what? Muscle for the contraction. All right. Let me be here. Okay. So I was talking about muscle contraction. See, I told you there will be a line like this. This is the Z line, EZ line. See over here, Z disc, it has been written. Okay. In between, there is myosin, and these small ones, the small thin ones are actin. Hope I am clear and I am visible to you guys. Okay. Now myosin and actin. Now there has been questions on this. This M line, A band, I band. These are very much important. Okay. So between this Z, okay, please pay attention. See, where you see only and only myosin. See, this is myosin, completely myosin. Am I right? Okay. Now see, these myosin are not just present like this. If I am drawing this myosin and if I am drawing actin, they are not attached like this. Okay, they are attached like this. Let me just show you. They are attached like, see, this. If this is the myosin, the actin will be almost still here. Okay, 2 actins over here, and here also actin will be almost still here. So you see one thing, you see that this is how they are attached. So for example, this is myosin, actin cannot be here. Otherwise, how they are going to, attach with each other and contract? Actin should be somewhat placed over here. Okay, so they can contract like this. All right. So question number 1 over here, that the band that consists of mainly, let me draw with this small pen. The band over here, that is concluding of some part of actin, whole part of myosin, and again some part of actin. This band is known as which band? A band. All are these questions, A band. Okay. If I draw a central line from these myosin filament, what is going to happen? This line is called as M line. This is called as M line. It is written over here, M line. Okay. So see, this is the thick filament. This is the thin filament. This is the thin one. This is the thick one. Okay. Actin is the, okay. All right. Then one more band is H zone. What is H zone? Look over here properly. H zone, just check over here. H zone is just comprising of only and only what? Myosin. Only and only myosin. So let me just draw over here with a different band. That this time we use black. So see, this band over here, that is just having myosin. So that black band that I have drawn, that will be called as H zone. What is going to happen? H zone. Now see what is going to happen. Let me draw you one more time. This is myosin, and let me draw actin. Now if contraction happens, actin will be completely like this, covering completely. It will cover from both this side, from both this side. Okay, it will become like this. Are you getting my point? Are you getting my point? Okay, so this will become completely like this. Okay, because of the contraction. See, something is in between and two people are trying to catch it, so it will completely overcome it. This will lead to, so during the contraction, which band should disappear? Can anyone tell me? Is it the M band, that is M line, it is getting disappeared? Is it the A band that is getting disappeared, or is it the H zone that is getting disappeared? Please respond to me. I have taught you just now which zone will be disappeared during the contraction. If they join completely like this, again, let me be very clear. Please respond in chat. That is how it will be very much interesting. Okay. See, if I am saying that this zone having complete myosin, this zone having complete myosin is called as what? See, you are not still not answering. I think am I not being that clear? Okay, so answer is H zone. Okay, H zone. Why H zone? Because H zone was the part where only, yes, someone has answered, I think so. Yes, H zone. Very good. Arshad, very good. That answer is H zone. Okay, so hope I was understandable. These are the questions that has been asked before. Okay, so be very clear with this. Okay, now let me go take you through this particular diagram. Okay, you can see this diagram. Please look over here. What is going to happen? First of all, okay, have you heard of troponin, tropomyosin? Okay, hope you have heard of it. See, tropomyosin is something like that is covering this. Over here in my diagram, this is myosin. The blue one with the blue one is actin, and there is something border. Some border is like that. This color particular. This border is called as tropomyosin. It is doing what? It is trying that actin and myosin should not, should not bind like this. It is like a gatekeeper. Okay, it doesn't want the contraction to happen. Okay, so if I want to contract the muscle, what I have to do? I have to throw away the tropomyosin, and then only I can interact with actin and myosin. All right, so let's see what are the steps in the contraction. The impulse of neuromuscular junction will cause release of acetylcholine. Yes, definitely this is very important. That will lead to depolarization and lead to calcium from the sarcoplasmic reticulum. Now this calcium is going to do what? It is going to bind with troponin. Okay, and by doing so, it moves the tropomyosin away. Okay, tropomyosin has been thrown away. Now the site is exposed. Okay, the site is exposed now. Troponin and tropomyosin bind to the calcium, which expose the myosin binding site. So hope you guys are understanding. If this was the myosin binding site over here, from the maybe red pen I can use. Here was the myosin binding site. Okay, this red one. Guarding that, there was a person known as a bad person known as tropomyosin. What happened to that? Just calcium came and tropomyosin was thrown out. Okay, tropomyosin was thrown out, and with that, what happened? The binding site got exposed, and now actin and myosin can interact. During that, what do we need? We need ATP. Okay, ATP breakdown occurs and myosin is pulled. So this is how it has been done. And also while relaxing, also the same thing happens, vice versa, and it should be again there will be ATP used. So in both this method, we have used ATP. So this was all about muscle physiology. See, there is one more topic in muscle. If you want to study type 1, type 2, and type 3 muscle filament, you can study from QRS if you want. It is at present out of my scope.